Okay, so I want to talk about the mode of action of the pesticides. Yeah, obviously, we are not going to cover all the pesticides. We will just pick a few which we think are important or which we think are representative of this mode of action. So let me start to present. So the topic is mode of action of pesticides. Now, in understanding of the mode of action, maybe is based on the biochemical and physiological processes that lead to the destruction of the pest when it comes into contact with the pesticide. We are going to consider the following pesticides: organophosphates, carbamates, organochlorines, pyrethroids, and the triazines. Now, the organophosphates are widely used to control insect pests. They've tended to replace organochlorines because of persistence However, we have frequent sprays when using organophosphates during the course of a growing season. Organophosphates are very effective on aphids. Now, on the chemistry of organophosphates, we can represent them like using this structure. Phosphorus is the key element, and it is joined by what we call the methyl or ethyl groups, represented by R10, R20, or here or is representing oxygen. Y could represent oxygen or sulfur. X could be a specific organic group. So basically, this is the structure of an organophosphate. For example, this is structure where we have P attached to oxygen or sulfur. It's also attached to either the methyl group via the oxygen, and we've got this X, which is another structure. So this is the basic structure of an organophosphate. Now, organophosphates have got two other groups, which are usually methyl or ethyl. These are the other groups. These are the other groups I am referring to. X represents a complex aliphatic group. It is sometimes referred to as the living group, this X part. So organophosphates can have this link POX, this is oxygen, or PSX, this is the sulfur. Right. The first group we may look at for organophosphates, we have this structure here, where we have P attached to 4 oxygens. But here we are having these other groups represented by R, and also another group represented by X. But the 4th oxygen has got a double bond here. Now, this structure is common with any one of the following organophosphates. Number 1, we have got chlorvinphos. We also have another organophosphate called dichlorvos. The other one is mevinvos. The other one is phosphamidone. These are orthophosphates. Orthophosphates because of this oxygen here. We also have what are known as the thione phosphates, or phosphothionetes, or simply thione phosphates. The difference is this S. We have got the orthophosphate. If we put a sulfur phosphates, and the group within the thione phosphates, we have got a bromophos, diazinon, phenyltrione, parathione, then primiphos, methyl or ethyl. Then sometimes you have got a sulfur at this base. This is now a thiol phosphates. Thiol representing sulfur, or phosphorothiolates. Some of the examples are dimethyl methyl, oxydimethyl S-methyl, and vamidothione. It's because of this now S. And this group, which have the living group just attached to the phosphorus. These are phosphonates, and we've got examples like triclophone, butonate. These are the 2 examples. And the last one has got this structure. Pyrophosphoramides. Shradan is one of the insecticides. So you can see there are a variety of these organophosphates. I mean, with the time, you'll get to know them. Now, here is a group of enzymes which can attack organophosphates in the insects or humans. We have a This should be phosphatases. So we've got phosphatases or type A esterase. The other enzyme which we have is microsomal monoxidase or simply cytochrome P450. We have also carboxyl esterase, which act on the organophosphates. We have also glutathione S aryl transferase, then glutathione S alkyl transferase. So these are some of the enzymes which catalyze organophosphates. But I'm sure you may remember this cytochrome P450, because we are going to talk about it a bit. Even the phosphatases. Here I'll put a structure just to show you where the enzymes act. If you have got this O and this ethyl group, a carboxylase esterase, because there's an est. If we put a nitrogen here, this becomes an amide. And the result of that is that we have an enzyme called carboxylamidase, which can facilitate the breakdown here. We also have microsomal monoxidase, which can affect this amine group. So let us move on to the mode of action of organophosphates. I have tried to give you a range of these organophosphates. Basically, organophosphates inhibit an enzyme either in humans or in the insect called as acetylcholine enzyme or ACHE. This enzyme resides in the nerve tissues and the neuromuscular junction. Neuromuscular junction is the point of contact of the nervous system to the muscles. Now, this enzyme has got a high affinity of binding, and this can inhibit it. Come in. Sorry. Yeah, good afternoon. Yeah, just put them on the. Yeah, this one is the second file after the blue one. Yeah. Okay, so ACHE, which is the enzyme, it has the responsibility of destroying the neurotransmitter known as acetylcholine. So within the nervous system, we have this acetylcholine enzyme, which has to destroy the neurotransmitter known as the acetylcholine, ACH. Because ACH is a function of transmitting messages. Once the message has been transmitted, it has to be destroyed. We also have choline esterases, which are hydrolases that can catalyze the breakdown of ACH through the acetyl transfer. Now, if we look at the rates of breakdown or rates of hydrolysis in the reactivation of acetylcholine enzyme following its reaction with a pesticide, which we call phosphorylation of the active site. That combination of a pesticide plus this enzyme is very slow to break down through hydrolysis. And because of that, the enzyme may not be released. So if you put a pesticide, the pesticide combines with the acetylcholine enzyme and forms more or less a permanent structure. But without the pesticide, the normal combination of acetylcholine enzyme and the neurotransmitter acetylcholine would usually break down via hydrolysis. And this will enable acetylcholine to carry out its function. Because once it has transmitted a signal, it needs to be broken down. Here I'm just giving you a structure of a nervous system where we have this, what we call the axon, being attached to the muscles. And acetylcholine plays an important role in transmission of the messages. So you can have, this is the nervous system. And within this nervous system, we have acetylcholine, which transmit messages. Now, where you have two where you have two groups of structures of the nervous system, we have got what are known as the synapses. Now, it is at these synapses where we will talk about acetylcholine transmitting messages. Here I'm just putting these structures of the nervous system so that you have an appreciation of where these things are taking place It could be inside an insect or a human being. Here I've got a diagram of the synapse or the junction of the nervous system. On our left here, we have got acetylcholine represented by that black box. And here we have got the acetylcholine receptor. So what happens is that acetylcholine will move and it inserts itself at one of these sites and it gives a signal. Once that signal has been given, this acetylcholine is to be freed so that the signal is actually stopped. So what exactly happens is that you have acetylcholine, that box, and that star, which is the enzyme, the other, so that the signal has to be passed on. After that, this acetylcholine has to be liberated so that it can be reused. So each time it has to combine with the enzyme, but temporarily, then it has to be released. So the importance of the enzyme is to break continuation of this message. Now, if we use a pesticide, the pesticide is similar to the acetylcholine enzyme. If it is an organophosphate, it will combine with the acetylcholine, because the acetylcholine is the transmitting substance. Once it has joined the acetylcholine, this is more or less a permanent feature. So there's no breakdown like what is happening here. So it's a permanent feature where you have this star and that small box combining permanently. So what happens is that the messages are always being fired at the synapses. So there's continuous transmission of the message. And it is the continuous signal transmission which eventually kills the insect. Well, probably this may not be clear, but this is the acetylcholine, this is structure. And this other structure here, which looks like a string, and at the end there's SER there, is that and OH. This is the end of the enzyme, which will combine with this other end of acetylcholine. Once at the second stage, there's now a bond, and acetylcholine is actually released. But this, the choline is released. But this part here is not permanent. Later on, it will break off, and acetylcholine is actually regenerated so that it keeps on combining with the enzyme after the message has been transmitted. But if we put an organophosphate, at the end here, we create a permanent feature. And of course, part of the organophosphate is released. But now that this is a permanent feature, it means that the message will keep on being fired, and that will kill the insect. So this is another version of looking at it. Or you can try to use this diagram or this one. This is a dramatic way of trying to probably make you understand what is happening at the joint where we have the nervous system touching each other, the synapses. So we have the organophosphates are said to be ACE inhibitors or acetylcholine enzyme inhibitors. So what happens is that if you inhibit the acetylcholine enzyme, you actually increase the level of acetylcholine, which is the transmitting substance. The acetylcholine will transmit signals in the synapses. So there's overstimulation in the synapses. This can cause, some symptoms of organic poisoning. And you are saying this is happening here. That is where we have the synapses. And this is probably our enzyme, ACHE, and this is our organophosphate, which is being demonstrated like this, which is actually killing the ACH. And as a result, acetylcholine is accumulating, and the message is being passed on continuously, or it causes overstimulation, which will end up killing the insect. It's just a demonstration of what is happening, probably to make you understand. Again, these are some of the effects of organophosphate poisoning. Where it can weaken muscles, causing all sorts of things, which you can read on the slide. We move on to carbamates. The carbamates are derived from carbamic acid. And we've got what is known as physostigmine, which is a naturally occurring choline esterase. It is present in cannabis bean or Physiostigma benosum. It's a bean which is found in West Africa. So the people in West Africa usually did what were known as witchcraft trials, where they actually forced the people to drink an extract from this bean, which is quite poisonous. If somebody drank that stuff and died, the person was labeled as a witch. But of course, the survivor of the accused can be attributed to more of or her ability to vomit than the extent of guilt. So excess of victims. So after eating this bean, there was excessive acetylcholine, which ended up killing people. So these are some of the insecticides known as carbamates. You've got carbamic acid. We've got this carbaryl, carbofuran, and primicab. So these are the structures: carbaryl, carbofuran, aldecab, oxamyl, methomyl, propacate. These are some of the commonly used insecticides. Now, the mode of action again is similar to that of organophosphates. Now, these carbamates combine with the acetylcholine enzyme, but within the nerve tissues. The carbamates compete with the acetylcholine for the enzyme ACHE or acetylcholine enzyme. Now, excess acetylcholine will cause continuous transmission of messages, and this actually kills the insect. So the mode of action is similar to that of to those of organophosphates. But the carbamates can release acetylcholine faster than the organophosphates. Here, this is just a comparison of where we have acetylcholine without any interference. And this is the process which is very rapid. So we have the acetylcholine enzyme combining with the acetylcholine. Then afterwards, this can actually break very fast. But if you put a carbamate, the carbamate becomes part of the system. And this is a slow process to break down. This is what can kill the insect. It's even slower when you are talking about organophosphates, which are combining with this enzyme. So after this union, the breakdown is a bit slow. or it may be almost permanent. Here, the enzyme is quickly freed. So this is the situation without a pesticide, where we just simply have acetylcholine. Then we can substitute acetylcholine role with the carbamates or with the organophosphates. Now let us move to organochlorines. Some of the most widely used insecticides in history are members of the organochlorine class, and they include DDT, dieldrin, aldrin, hepatachloriden, linden, and toxaphene. So this is DDT, this is dieldrin, this one is linden, this is heptachlor, this is Mirex. Now, DDT has got neurotic effects. Now, the DDT affects the peripheral nervous system. It can act on the nerve muscle system. It destabilizes neural activity. The result is that you have got a leakage of sodium ions through the nerve membrane. It is this leakage which creates a destabilizing negative after potential. So the presence of DDT will cause leakage of sodium from the nervous system. As a result, the insect becomes excited. So it is this excitement which eventually kills that insect, or what you can call hyper-excitability of nerves and muscles. Come in. Sorry for that disturbance. So we're talking about the mode of action of DDT. Here I'm just showing you the effects of poisoning using DDT. Now, if we're going to measure the nerve transmission, in a normal situation, we get this top graph. But when the insect becomes poisoned, we get all these various peaks, which shows a disturbance, which means the insect becomes too excited and eventually it gets killed. We also have pyrethroids. These pyrethroids are obtained from Chrysanthemum cinearifolium, which is a plant grown in Kenya and Tanzania. Sorry. pyrethroids can be synthesized from these, can also be synthesized apart from being extracted from this plant. So these are the structures of pyrethroids. They have these structures. Again, the mode of action of pyrethroids is similar to that of DDT. We have the nerve excitation, which occurs as a result of changes in nerve membrane permeabilities. In fact, all these two pesticides, pyrethroids and organochlorines, they cause the nervous system to become very permeable. And this can influence the movement of sodium and potassium ions. One of the most dramatic things which they cause is a rapid knockdown effect. They have a quick effect on the target insect. They have a neurotoxic action, and the effects resemble those of DDT. They cause hyperexcitation and death. They cause a lot of excitement in the insect. Sodium and potassium conductance are altered when the nerve is poisoned. So you have the potassium efflux stage of recovery after passage of an impulse may last much longer in the pyrethroid poisoned than in normal action. This is why it causes this hyperexcitation. If the impulse or if the signal was measured, the ones which passes through a nervous system, in a normal situation, we have this kind of signal. But in a poisoned environment, you get all these, close together, these peaks showing excitation. Some have done experiments which show that this excitation depends on the environment. It depends on the temperature, which is another thing. But otherwise, where you have the control, no pesticide, this is where we have the pesticide affecting the nervous system. If I could just summarize the action of organochlorines and pyrethroids, they can affect this part of the nervous system. Normally, they are affecting the movement of sodium and potassium, which influence the signal transmission. On the other hand, organophosphates and carbamates cause excessive acetylcholine to accumulate, causing the transmission of messages to be continuous, which can lead to the death of the pest. Here I'm going to talk about the mode of action of triazines. This is totally a different environment. We are talking of the herbicide which is going to kill the weed. Triazines are mainly applied herbicide, soil-applied herbicides, but it can also be used as post-emergence herbicides. Atrazine is one of the commonly used herbicides. Ametrine is also another triazine, which can be used as a post-emergence herbicide in sugarcane. And these herbicides have got a potential to be persistent. So these are the triazine herbicides. Atrazine, ametrine, simazine, propazine, and so on. So let us focus on the mode of action of triazines. Now, for atrazine, the focus is on the photosystem 2. Probably I'll show you what is photosystem 2. Normally, we have this chlorophyll A and B and carotene, which, when struck by light, this region gets excited. Also, this other region, P700, gets excited. Now, when electrons leave 8, they go to the primary electron acceptor. Then they go to plastoquinone, cytochrome complex, plastocyanin, and eventually they are taken by P700, which is also chlorophyll A and B, plus carotene. Now the electrons will also go to the primary electron acceptor, and eventually they go to ferrodoxin, leading to the formation of NADPH. So on the one hand, we have got NADPH being formed here, and also at this point, we have got ATP being formed from ADP. So if we apply atrazine, atrazine will come somewhere in between here. The electrons, instead of going to cytochrome complex, they will go to atrazine. Once they go to atrazine, it means they won't go through this route. Once they don't go through this route, it means this NADPH won't be formed. It also means ATP won't be formed. But what may also happen is that there could be an accumulation of hydrogen peroxide, which will start to burn the plant. And at the same time, that plant will be starved of ATP and NADPH, which are required by the Calvin cycle, which means the Calvin cycle will be slowed down. You know, Calvin cycle produces carbohydrates. I mean, there's a whole range of things which will happen to the plant as a result of this disturbance which happens here. That will kill the plant. So this is what we wanted to explain. We are saying that atrazine may take the electrons from photosystem 2 between primary electron acceptor and probably plastoquinone. Then we have a formation of peroxides, which will kill the plant. Of course, this nitrate reductase activity can also be increased. So what we are saying is that the pathway of the photosynthesis is actually blocked by atrazine. Once it's blocked, then fixation of carbon dioxide is also affected. So ATP generation is affected. And the dark reaction of photosynthesis is also affected. We know the dark reaction is the one which is responsible for the production of carbohydrates. And this oxidation can also affect the plant, leading to plant death, because atrazine will be diverting electrons from the electron transport system. So this is what we wanted to explain. It's either the atrazine is here or it's there, and it blocks this whole system. So here is the summary of the processes. We've got the mode of action of the following pesticides, which we have covered, which is organophosphates, carbamates, organochlorines, pyrethroids, and atrazine. We know there are a lot of pesticides which we can cover, but we think these represent some of the important groups with the mode of action which is similar to common pesticides. So this is it on the mode of action of pesticides. Let me stop sharing. Hello?
Hello.
Any questions on this one?
None for me.
Is it clear?
Very clear.
I've given you 2 areas, in the nervous system where pesticides can act. But for the herbicides, we know there are quite a lot of, examples. We've just chosen the triazines.
Okay.
Yeah. Anyway, the idea is that you should have an idea about the mode of action of a pesticide. Let me stop recording.